start by thanking the VFS team for the organization and the Hungarian local team also for managing this meeting. I think we'll need to keep our schedule and get started. I hope everybody will be squeezing in. And we'll just start without further ado with the I think most interesting session of the whole meeting on the health effect of radon. It's on. Yeah. So and Laura from Barcelona, Laura Mezquita, please.
Okay. So, I can be here.
Yeah.
So hello everyone.
Maybe stand behind the thingy.
Yeah. So good morning, everyone. So my name is Laura Mezquita. I'm working at Hospital Clínico Barcelona, and I would like to thank the organization to invite me to present all the data that we have been working during these 5 years and to talk about the molecular characterization of lung cancer associated with radon. So for this, during these 15 minutes, I will start with a brief summary about the clinical perspective of lung cancer, to then discuss what is important for us about the molecular profiling of these tumors associated with radon, the results from the task 4.5 challenge, future perspective, and a few take-home message. So the thing is that we are working on lung cancer, which is a big public health issue. Is the leading cause of cancer mortality around the world. The prognosis is improving, but even though there is, so it's really far from other solid tumors, and this is because the majority of the cases are diagnosed in advanced disease, where the disease is not curable. So this lung cancer, obviously the most important risk factor is smoking, but this is not the only one. It's a multifactorial disease, and in clinics we have this situation. There is a stigma associated with smoking and tobacco, and there is it's completely silent radon. So we are not the typical, so I'm a medical oncologist, so we are not the typical medical oncologist that usually is not known the other risk factors. So we don't know really which is the clinical impact. Just to mention how lung cancer diagnosis it works. So initially the patients develop some kind of symptom because of the primary tumor of the metastasis. So we have this imaging with a lung cancer here, and then we perform different tests to evaluate the extension of the disease, the biopsy to confirm the diagnosis, and then we classified according to the type of cell, the histological subtypes of lung cancer in non-small cell lung cancer or small cell lung cancer. And in non-small cell lung cancer, these subgroups that I will mention later, adenocarcinoma, squamous, and other subtypes. So thanks to this radiological examination, we stratified the patients according to the stage. So the most common is stage 4 with metastasis. And then according to this stage, we decide the treatment. So in early stages, the main treatment is surgery or radical therapy, concurrent chemotherapy, chemoradiation, and in advanced stage is systemic therapy, because it remains as one of the most important treatments. Systemic therapy is all the treatment that can arrive all the parts of the whole body to treat not only the primary tumor, also the metastasis. And for this, we select the treatment in patients according to the tumor biopsy. So for us, it's really important, the tumor biopsy, because we classify the patients, according to the histological, molecular, and immune profile, according to this tumor biopsy. And for us, it's important because this information is mandatory before the treatment selection. And the treatment selection is based on molecular data that I will discuss, because we classify the patients according to the molecular profile of the tumor, and according to this molecular profile, we guide the treatment strategy. Better? No? Ahora? Better. No. So according, these biomarkers are mandatory. So we have this we name precision medicine. We classify this patient, the tumor of the patient, according to molecular and immune mechanism, and decide which is the best treatment for the patient. So the question is, any of these profiles, so histological, molecular, or immune profile, can be associated with radon, and obviously the precision medicine, because it's a combination of all of these biomarkers to modify the treatment and also the outcomes of the patient. So we try to demonstrate this because we have some information in smoking, but there is no information about radon or other risk factors. So indeed, we know that radon smoking is associated with poor outcomes in patients with lung cancer to continue smoking after the diagnosis. It's associated also with better response to immunotherapy, so can influence the smoking, the response to therapies, to immunotherapy, or to poor response to other therapies, the targeted therapies, but we don't know what is the role in the case of radon. So we have this information about the pathological profile of radon. It's associated with all the histologies, adenocarcinoma, most common in non-smokers, small cell lung cancer in smokers, but about the information of the molecular immune profile, we have not a lot of data. We have the main data is in DNA repair mechanism, but there is very limited from the clinical perspective. So we have all the solid evidence in cancer risk, but we have very few data about the mechanism. We have no data about lung cancer profile, and we have no data about the impact on the patient outcomes, so the survival and the response to therapies. And this is important because based on this, we need to develop more dedicated research with this focus, which is the precision medicine, because all of this modification influence of the tumor, of the radon, in the tumor, can help us to identify vulnerabilities, and based on this we can even develop targeted therapies or immunotherapies in the future based on certain biomarkers that we can identify in tumor associated with radon. So our first area of interest was these early clonal alterations, because there are mainly mutation, fusion in the tumor associated mainly in non-smokers, in which radon is one of the main risk factors. And we developed different prospective small cohorts evaluating, and also large ecological studies demonstrated an association between the prevalence of certain driver alterations in these patients and radon-prone areas. So based on this, we built these 3 tasks to evaluate the molecular profile of lung cancer in these 3 different models in rats, from the French Atomic Energy Commission, in uranium miners, and also in a prospective cohort, which is the EORTC lung cancer group by radon study. So initially, we tried to sequence 52 samples, but at the end, we had only, because of the quality of the quality control, 36. Thanks to this collaboration with the CIA, we observed that there is in rats a correlation with all the different histological subtypes. Also, in cancer pathways, the most important was RTK-RAS, which is super important because in this pathway are all the oncogenic driver alterations that are important for us, and indeed we identify some of them, MET, RAS, and NF1. Also DNA repair pathways, not non-homologous enjoining that we aim to identify, so we identified mid-batch repair and base excision repair, but indeed there was a super high complex of different variants. And we were also interested in the fusions, because the fusions, is a hot topic in ionizing radiation, and also based on our previous data that we identified that ALK fusions were most commonly associated in high radon areas. So numerically, we identified high number of fusions in the cohort of radon exposed to high and prolonged exposure. And when we evaluate indeed the type of fusions, we identified 2 important points. One, we identified fusions associated with ionizing radiation reported in other tumors. So indeed BCL11B, which is a fusion identifying leukemia associated with gamma radiation. And this is something that we are now characterizing better. And also we identified oncogenic fusions, including ALK, that was exactly the same driver that we identified in the ecological trials. So we know that this is low level of evidence, but even though it's hypothesis generating, and this is data that can evaluate. Even though this is RNA data, it's not protein level, and we need to confirm that the data that we have in RNA is also present at protein level, because of the, it's too long to explain, but we need to have the protein level, And indeed, it took a lot of time to optimize the biomarkers, this immunohistochemistry, because it's not validated for rats. And finally, we identified that there is a correlation between RNA data and our protein level data. Our immunohistochemistry demonstrated that at protein level data, we are seeing also this overexpression. We were also interested in the mutational signatures, so this is big papers demonstrated the different mutational signature associated with all the carcinogens that you can imagine, also air pollution, but there is no signature associated with radon, so we were super interested on evaluating this. And we define SBS, DBS, indels, the different copy number, it doesn't work, but single base substitution, there is a predominance of CT. It's too long to explain, but it's mainly associated with DNA repair, it's associated with paraffin preservation, that can be also a limitation of these samples, because these samples were in paraffin, but it's also associated with other signatures in ionizing radiation. So all of this is generating CT. So it's difficult to know what is exactly each of these. And then DBS also, if you see all the different signatures are exactly the same regardless the radon groups. So I mean, regardless the type of dose and the pattern of exposure as the same signature. This was not the objective of radon, but we were super interested also in the immune profile, because it's together with the molecular profile. We should not separate both because are in balance. And indeed, we expected to identify an inflamed tumor microenvironment associated with more T-cell activity, so with more response to immunotherapy, similarly to smoking. But indeed, we identified a high proliferation of immune cells, but not the T cells. We are identifying more macrophages and other immunosuppressive cells that can explain why these patients and non-smokers didn't respond to immunotherapy. So we have been also validating these results with the protein, so in that at protein level, low TILs and low density of T cells in the tumor microenvironment of these rats. Another important point in immunotherapy is the TMB, because the tumor mutational burden is the number of mutations per million base is a biomarker of immunotherapy. So in smokers associated with chronic exposure to carcinogens is associated with high TMB, high number of mutation. We expected exactly the same for radon, and indeed, as you can see here, this is all the TMB tumor by tumor in the TCGA cohort, and this is a high TMB in the cohort of rats. Finally, we evaluate the carcinogenesis pathways and the role. So this is the cancer pathways, the upregulation. I saw this in another annual meetings, just to show how complex can be all the interaction of different cancer pathways. But what is something new is that we evaluated the chemical carcinogenesis pathway that was also upregulated, showing that each of these is one carcinogen, group 1 carcinogen, that is also upregulated in radon, showing that there is this potential and synergistic effect between the different pathways and different carcinogens. So this is something that we need to explore furtherly. The second cohort is minors, thanks to Maria, collaboration with Maria and BFS, initially 70, but finally 30 analyzed, 3 cohorts, high, intermediate, and low, high radon associated with high number of variants as expected. The problem is smoking is missing in 30% of the cohort, so we can identify different findings that can be also associated with smoking and not only to radon. In general, genes identified that are commonly associated in non-small cell lung cancer, and particularly in adenocarcinoma, to emphasize MUC4, which is an oncogene associated with the activation of the HER2 pathway, which is super important in our tumors, and DNA repair, homologous recombination, and DBS. Exactly the same pathways as in rats, observed in minors. In comparing low versus high, 3 genes, MUC4, MUC6, and also a gene related with the degradation of the stroma, associated with poor outcomes, were the genes more commonly identified in higher radon group, and in the driver groups, the BCL11B, which is one gene that we identified as rearrangement in rats, we are identified a higher number. It's impossible to explain all of this in 15 minutes because I'm very super complicated course, so sorry, because I need 5 minutes more to explain, or I will finalize now. But the thing is, the thing is that we identified BCL11B, which is rearranged in rats, and now we are identified a high number of variants associated with the same gene. This is the gene that I said that was associated with leukemia induced by ionized radiation, gamma radiation, so this is something to explore, and we are trying to validate this in protein in patients. And also HER2 that I mentioned before is important because of MUC4 and NRG1, which is a ligand of HER2, which means that a HER2 family is something that we need to explore furtherly. We have no RNA data, but we try to use new tools using new tools that can extract information of the fusions using the exome data. These chromosomal aberrations, we identified a lot of deletions, also rearrangement, and in cancer, in genes that are interesting from the ionizing radiation point of view, and also from our clinical point of view, Notch and Red. TMB is high, particularly the combination of radon and smoking together. And mutational signature, again, the same that in drugs, in CBS, and also we identify similarities with ultraviolet. Okay, so I find it. I cannot.